This recorded session is dedicated to the second half of our lecture for module number 4 on antigens, antibodies, and antigen-antibody interactions. We will have this lecture entirely for antibodies and antigen-antibody interactions. Um, Before we proceed, I will be dividing this session into 30-35 minute sections as per your request, since in order for me to easily upload an entire video covering the whole lecture, and also to address your requests, wanting to have, or since most of you expressed that they want to have the videos in 30-35 minute short video formats. So to meet halfway, I will be providing one video, and then I will add 10 to 15-second breaks probably every 10 slides, no? So that you can also pause the video as you listen and take down notes. Okay. So last synchronous meeting, we described the entirety of antigens. So I walk you through some important concepts on the biochemistry, the physiology, as well as the behavior of antigens once that, especially they are already inside the animal body. No? So before we be able to appreciate more about how antigens and antibodies interact, we need to, of course, understand what antibodies are. No? We also have to study the biology of antibodies. Biochemically, antibodies, also known as immunoglobulins, they are glycoproteins. So you know from your basic biology that a glycoprotein is a biomolecule that's composed of proteins and carbohydrates, no? Oligosaccharides. If you take a look at the chemical structure of antibodies, no, you will see that there are, or you will appreciate the presence of oligosaccharide chains on the sides. Not really making a substantial contribution to the whole antibody protein. No, so your oligosaccharides, you expect them to be present on small portions. No, they only act as side chains on um, mostly on the on the crystallizable region of the antibodies. So antibodies, they recognize, in terms of functional, they recognize and neutralize microbial toxins and foreign materials. So both pathogenic cells and their byproducts that are considered as antigens, as foreign to the animal body, can be recognized by immunoglobulins. No? One type of antibodies are membrane-bound antibodies. They act as receptors for antigens on the surface of our immune cells of B lymphocytes. And the second type, another type, the non-membrane bound immunoglobulins, they are involved in the inhibition of entry, prevention of entry, and actually not prevention, but more of inhibition of the continuous progress of infection and the spread of the pathogens that are found in blood, as well as in connective tissues. So your non-membrane bound, they are secretory antibodies. You expect them to be in the circulation, in the blood. Some of them may stay in connective tissues, and um, if a pathogen breaches the anatomical barriers of the animal body, they're one of the first line of defenses. No, they're one of the first immune components that will go ahead and make the prevent the interaction between, or prevent the further progression of infection. No? Okay, so let's further discuss about this membrane-bound antibodies and secretory antibodies. No? Membrane-bound antibodies possess transmembrane domains that anchors them into the lymphocyte's membrane. So you remember B cell receptors. No? So B cell receptors are actually membrane-bound antibodies. They are antigen-specific. They as antibodies that are anchored on the B lymphocytes cell membrane. They act as antigen-specific receptors. And the reason why they can do that is because they have transmembrane domains on their on their structure. And on the other hand, unlike membrane-bound antibodies, the secretory antibodies, they do not have that transmembrane domain. And though, if you take a look at the biochemical structure, structurally the same, no, with membrane-bound antibodies. The only difference is that they don't have the transmembrane domain, allowing them to, or giving them the incapacity to bind to the cell membrane, no? As an important element in humoral response, no, these antibodies are primarily produced by our B lymphocytes, by our B cells. However, we know that a few recent studies already recorded and detailed the production of some types of antibodies by other cell types that are actually non-lymphocytes, no? Like skin cells. Remember the one, if you're doing your enabling assessment for this module already, you've already encountered the idea using the assigned paper or via the research paper I assigned to the class for synthesis. So you have encountered the idea of immunoglobulins produced by non-lymphocytes. No? So going back to antibody generation by our B lymphocytes, no? Initially, our pre-existing B lymphocytes that are in a resting state are expected to encounter antigens either by themselves alone or through the help of other immune cells. No? So eventually, these stimulated B cells that have already encountered the antigen, so the epitope of the antigen is already recognized as foreign by our membrane bound or by our secretory antibodies, no? And for the case of membrane-bound antibodies, to be specific on this illustration, once that they are encountered, eventually the B cells become stimulated, no, and they will give rise to our antibody-secreting plasma cells, no. These antibody-secreting plasma cells will the maturation or the maturation of these plasma cells will depend on the antigen that stimulated them. Okay, let's take a look at the basic structure of our antibody, since we've been talking about much on how important it is, some intro on its biochemistry, and some if discussion on the different types of antibodies that are present in the animal model. Now, for immunoglobulins, no, although there are different immunoglobulins that can differ structurally. So, because actually the types of immunoglobulins present in higher vertebrates in animals with backbone, there are a numerous, or there's a set of immunoglobulins that are present in animals. So although they differ structurally, they can all be described to be built from the same monomeric unit, the same basic unit. No? Antibodies are Y-shaped protein structures, Y-shaped glycoproteins that have four polypeptide chains as their basic unit. So consisting, the four polypeptide chains are two identical light and heavy chains. So here we can see our light chain here. It's one, two, and then we also have our heavy chain. This one, the one represented by the green structure. Okay, so our light chains, they weigh around 23 kilodaltons, while the heavy chains are within the range of 5 to 70 kilodaltons. Okay, now let's look at the structure of immunoglobulins in detail. An antibody an antibody's monomer can also be seen as having two separate regions. So aside from the fact that, or aside from considering the chains as identified components, we can also take a look at the antibodies based on two separate regions. No? A fragment antibody binding region, a FAB region, and an FC region, or a fragment crystallizable region. The fragment antibody binding region is a portion of the antibody that has the capacity to bind to the antigen and actually consists of one variable and one constant domain of each of the heavy and light chains. So here, let's take a look. So one variable for the light chain, one variable for the heavy, one constant for the light chain, and another constant for the heavy chain. No? So this comprises our fragment antibody region. And you can see on its ends, actually, we can appreciate the antigen binding site, so the paratope, where the epitope of the antigen is expected to be chemically attracted to. No? Now, the fragment crystallizable region, on the other hand, is the distal region of the antibody. Okay, and it is composed of two identical disulfide linked peptides containing the heavy chains CH2 and CH3 domains. So it's just a continuation of the heavy chain, but the domains CH2 and CH3, which are both constant, are comprising our fragment crystallizable region. This region communicates with some of the cell surface receptors called FC receptors, and this feature of the FC region, the fact that it communicates to FC receptors, help antibodies stimulate the immune system. So let's examine in detail yung the variable and the constant regions. The amino terminal variable regions that are found both in the heavy and light chain are actually responsible mostly on the on the antigen recognition process. And unlike the carboxy terminals, we expect at these ends of our light and heavy chains, no, ito yung amino terminal, while the ones that are proximal to the fragment crystallizable, the ends of the light chain and the heavy chain that are within the FC region component are the carboxy terminal. So the carboxy terminal regions, on the other hand, no, they function for the effector processes of the antibody, such like for the cases that they are already bound to a B cell as a B cell receptor. No? So we expect that the carboxy terminal end of the polypeptides will work on the effector processes and will not really contribute much on the antigen recognition part. Both heavy and light chains are composed of the immunoglobulin domain, and you probably encountered this in your biochemistry already. So the immunoglobulin is essentially 3D. It's more 3D compared to other conformations of proteins. And in our light chain, in one of in our light chain, there is one variable region that is immunoglobulin in type. Then there's also our constant region that's also immunoglobulin in type. While for the what do you call this? While for the heavy chain, the variable region also comprises of one immunoglobulin domain, while the constant region, ano siya, no, might be 3 to 4. Unlike in light chain, isa lang yung constant region natin, which is an immunoglobulin in conformation. So just remember, or ano lang, to summarize, all of these variable and constant regions of the light chain and the heavy chain, all of them are immunoglobulin in terms of conformation. No? Now, for the production, no, I'm going to make a quick discussion about how these antibodies, or the light chains and the heavy chains, to be exact, how are they synthesized? No? Okay. So another important concept that we need to touch base on, I have to mention this. Like most other proteins, the heavy and the light chain are formed inside the cells, of course, inside the plasma cells organelles, particularly the rough endoplasmic reticulum. And proteins called chaperones are involved in the process of immunoglobulin genesis, as these proteins are required for proper folding or unfolding of the immunoglobulin heavy chains as in the process of assembly there is some the adaptability or flexibility that needs to be observed, no? Because as the folding occurs, there are chemical interactions that are occurring in a cycle, whether they are existing or non-existing. Therefore, your essentially your components of your antibodies, both the heavy chain and the length chain, the light chain, specifically the heavy chain, should be really flexible or adaptable. The assembly process will include stabilizing of both the heavy and light chains by linkages called disulfide bridges, okay, form between two cysteine residues. And aside from the disulfide bridges that stabilize the interaction, the chains themselves, the side chains of the amino acids comprising the chains will also contribute to the entire association of light and heavy chains. No? The whole process will occur in the endoplasmic reticulum, and then it will be followed by carbohydrate modification, which will be required at the end of the assembly process. So at the end of the process, your immunoglobulin molecules, they will be separated from the chaperone molecules, from the chaperone proteins, and they will be now transported or shifted to the cisternae region of the Golgi complex, so that the carbohydrate side chains can be added to them already. No? So let's take a look here. It's not really evident here, but these cyan-colored circles are the oligosaccharide side chains that are added after the assembly of the immunoglobulin polypeptides, no, of both the light and the heavy chains. Okay. Then after the carbohydrate modification, eventually the mature antibody will find its way into the plasma membrane, normally via vesicular transport. And membrane-bound immunoglobulin molecules still will stay on the surface of the cell, while those that are secreted will form part of the antibody pool in the blood, no? Or they may also seed other parts of the animal body like connective tissue. Now, the second component of immunoglobulins that we will talk about are the disulfide bonds. We're done with the light and heavy chain. Now, let's talk about the disulfide bonds. Disulfide bonds are of two types: interchain disulfide bonds, along with non-covalent interactions. They hold the heavy and light chains together. So here we can see interchain. A disulfide bond. Okay, this one. Actually, this is heavy, no? So this one. This is an interchain disulfide bond holding our light and our heavy chain together. On the other hand, um, oh, we have our intrachain disulfide bonds. Within each of the polypeptide chains, they can exist as stabilizers within each. No? Kaya nga intra means within the molecule, while inter is across the different polypeptide chains. So within each of the polypeptide chains, our intrachain disulfide bonds are appreciated as follows. So intrachain within each of our separate polypeptides. Okay. Now let's talk about since the essentially I mentioned everything about disulfide bonds as stabilizers. Okay, because they're very strong type of chemical bond. So their presence in our immunoglobulins allows the immunoglobulins, the antibodies, to be of good of stable conformation most of the time. Okay, now let's talk about our third component for immunoglobulins, the variable and the constant regions of the chains. Okay, so the variable region is represented here by the blue colored part, includes the N terminals, the N terminal end of our light and heavy chains. No? So the variable region here, both for one, the blue colored one for the heavy chain, and our green colored one for our light chain. So they are the N terminal sila. So N sila. While the ones that are found on the near the FC region are comprising our constant region, no? Yung the constant region here. Okay, the end portion of each polypeptide is carboxy terminal. Okay, um, then okay. So I mentioned this already. No? Antigen binding sites, let's take a look, is formed by the variable regions of both the heavy chain and the adjacent variable region of our light chain. Okay, so here, antigen binding site, while the distal portion will work on the effector processes. Now, here, we were seeing a more detailed appearance of our immunoglobulin molecule. No? So the disulfide bonds formed between the cysteine residues we expect to connect the light and heavy chains in the carboxy terminus of the light chain and the CH1 domain of the heavy chain. The association of the heavy and light chains will occur partly to non-covalent interactions between the uh, variable region of our light chain and the variable region of our heavy chain. Now, and also between the what do you call the constant region of the light chain and the constant region of our heavy chain. Okay, so two heavy chains of each antibody entity are connected covalently by our disulfide linkages. Actually, this special region here, known as the hinge region, it's very important as this one. Actually, if you chemically degrade an immunoglobulin, no? So this hinge region plays an important separation point, no? Between our FC region, the fragment crystallizable, and the fragment antigen binding. No? Okay, so in IgG antibodies, one type of immunoglobulin, one isotype of immunoglobulins, the disulfide bonds are formed between the cysteine residues on the CH2 immunoglobulin region, which are near to the hinge. And if we take a look, no, actually, I'll show it later. We'll have a discussion on each of the isotypes naman. So we'll be able to appreciate the presence of subtle differences, no, in terms of the chemical structure of each of the isotype. Okay? So further dissecting the variable regions, as we've mentioned about the constant region being uh, uh, important or associated with the, I don't know, with the binding of the polypeptide chains. Very important for stabilizing the interaction between the heavy and the light chains. We'll focus now on the variable region. We also call the variable region the complementarity determining regions, or the CDR. You will definitely encounter this now if you do further reading about immune structures. No? So these hypervariable regions, they are made up of the N-terminal half of the light chain and the N-terminal quarter of the heavy chain that will contain amino acid sequences that are highly variable. So they're highly variable. These hypervariable regions, they are in direct contact to the antigen's epitope. Therefore, you can consider them as the paratope of the antibody. Okay. Now, from our last sync session, we discussed that antigen recognition requires specificity of recognition by a dedicated antibody. And this specificity is attributable to the variable regions of the light and the heavy chains. So that variability, hypervariability is the reason why there are specific antibodies that will recognize specific antigens. Okay, in general, we expect that antibodies from different plasma cells, they will have different specificity. So if you have two different plasma cells that are capable of recognizing different antigens, therefore the antibodies that, or rather, sorry, let me correct my statement. So if you have two different B lymphocytes that have B cell receptors that have membrane-bound antibodies capable of recognizing different antigens, no? You expect that as they mature, these two plasma cells will produce antibodies of varying variability. Okay, kasi these two plasma cells will have antibodies that will recognize different antigens. No? Though the recognition is still an entirely different story. No? We will discuss that in a while. I'll provide 5 to 10, or I mean a 10 to 20-second break. Okay? Okay, so we're back now. Um, so as I mentioned, the specificity and binding is an entirely a different, different domain, a different story. No? So, uh, although though we have the specificity of antibodies and antigens, no, we also have to remember the phenomenon of cross-reactivity, which happens when an individual antigen binding site of an antibody is able to recognize more than one antigenic epitope. No? Let's illustrate this phenomenon of cross-reactivity with the following photos. No? So cross reactions, they arise because the cross-reacting antigen shares an epitope in common with the immunizing antigen, or because it has an epitope which is structurally similar to one on the to the one of the found on the immunizing antigen. So here we can see shared epitope. It's one possible pathway where cross reactions arise because we expect that the cross reacting antigen will have a similar epitope with the original antigen. So for this case, our cross-reacting antigen is antigen B. So it shares the same epitope with our antigen A. Now, for this illustration, on the other hand, if the what do you call this? The cross-reacting epitope has a structurally similar or the cross-reacting antibody has a structurally similar epitope, no, similar to the immunizing antigen. Not entirely the same epitope, no. Ano lang, portions of it, probably the key amino acids are the same. No? So we have your cross reactions, cross reactivity expected to arise, uh, even though not the entire epitope is shared, no? Not the entire sequence of the epitopes is shared. Even a few similar amino acids that are shared by the cross-reacting antigen can already elicit cross-reactivity or cross-reaction. Okay. All right. So now, if we take a look at the antigen binding site further, no, let's examine that because we've already mentioned how we relate the antigen binding site of our immunoglobulin with the constant region of the ano, no, um, I mean, sorry, not the constant region, but how we differentiated, no, the contribution of the constant region and the variable region of our light and heavy chains into the process of antigen recognition. Now, as mentioned, uh, the constant regions are connected to or are primarily associated with the non-antigen binding parts of the antibodies. The fragment crystallizable region. So antibodies, we divide into several types. The major classes which we will cover also in this lecture are the IgM, IgG, IgA, IgD, and IgE. So based on the actually this five no no these five types, the variable region, the variable regions are almost the same siya across. I mean, the monomeric units or the sequences almost the same. Syempre magkakaroon na ng differences on the key amino acids. What differentiates these isotypes from one another is there are five potential variations in the carboxy terminal end of the constant regions of the heavy chain. So here, okay, so here you have your constant region, the CH1, CH2, and CH3. So the amino acid sequence variation here is the reason why we have several types of our immunoglobulins, why we have several types of antibodies. And also aside from the carboxy terminal portion or the amino acid nearing the carboxy terminal portion of the constant region of our heavy chain, we also have the side chains, the carbohydrate oligosaccharide side chains. Okay? As a review, let's remind ourselves that the constant region determines the effector processes, the effector function, or the mechanism that will be activated by the antibody in order to destroy the antigen or the foreign cell. Okay? Now, let's move on to the fourth structure of antibodies, the hinge region. I made a quick mention about this a while ago. So here, this is the hinge region. And this is the region at which the arms of the antibody molecule forms that recognizable Y shape. No? It is, as the name implies, it is called the hinge region because there are some flexibilities in the molecule at this particular point. So we expect that if you actually what do you call this? If you let's say for example, no, you make the you subject your immunoglobulin to enzymatic degradation. Let's say you use an enzyme, no? Like for this case, we have papain. If you will be generating your Fab and FC fragments, they will break away from one another through the hinge area, through the hinge region. So if you have your papain enzyme, you will subject your immunoglobulin. It will digest it and it will break the immunoglobulin molecule in the hinge region. So you will produce your Fab region containing the antigen binding site here. And you have your fragment crystallizable region. It's called fragment crystallizable because it can easily be crystallized. No. So that's the reason why it was given the moniker crystallizable, fragment crystallizable region. No. However, if you will talk about immunoglobulins in general, if you will crystallize immunoglobulins in general, it will be difficult, especially there are a lot of 3D conformation in the Fab region. No? So your antigen, actually both, if you combine the antibody and the antigen together, the complex, it will be very hard to crystallize them. No? Okay, now we have our fragment antibody binding. Let's take a quick review. So our antibody, any antibody, is able to bind to a particular antigen because it has a particular combination of the variable regions of the heavy and the light chains. Different combinations of these variable region in both polypeptide chains can bind to different antigenic determinants. You remember our discussion about antigenic determinants, no? So number of epitope, number of key amino acids found on the antigen. What else? Also the presence of external components like what do you call those? Haptens and adjuvants. They assist or they contribute to the whole binding process. Okay. Now let's take a look at the fragment crystallizable region. The FC region mediates the effector functions or the biological activity of the immunoglobulin, especially after after it's already after it already bound to an antigen. No? So within this fragment, different functions are mediated by the different domains. So some of the functions or some of the specific uh, functions related to the domains of the fragment crystallizable region is complement activation, complement binding, binding to Fc receptors that are of different types among immune cells, and we also have the idea of placental transfer of antibodies. No? Okay, so here we have let's take a look. Let's have an illustration. Okay, here. Okay, so going back to the fragment crystallizable region, so some dedicated domains for different functions. For complement protein binding, for binding to Fc receptors that are found across different types of immune cells. No? So the Fc receptor, they can be found on certain cells such as lymphocytes, our B lymphocytes, our no, not antigen, our natural killer cells. Okay. Also, we have polymorphs. Polymorphonuclears can possess Fc receptors on their membrane. Therefore, your antibodies are expected to bind to them as well. And all of them, actually, these other immune cells now, the interaction of an antigen bound antibody with them will elicit cascade of immune instructions. And then we also have placental transfer, domains dedicated for placental transfer of the antibody to from the mother to the progeny. Okay. All right. So we've discussed thoroughly on the biochemistry of immunoglobulins and the structural biology. We've made quick mentions or discussions about them. Okay, we'll now proceed to the different isotypes or the antibody classes that are found in the human vertebrate. No? So we have a set of five isotypes. No? We have IgG, IgM, IgD, IgA, and IgE. So a good mnemonic here is the word gamed. So IgA, IgG, A, M, E, and D. So these antibody classes, they are different from one another in terms of form and of function. So to reiterate, the heavy chain's constant region is responsible for the determination of the biological activity of each of the antibody class. And from this illustration, we can appreciate early on the differences, no, in the constant regions of the heavy chain. So you can see on the number of domain. Okay, so I mentioned a while ago that the constant region of the heavy chain can be 3 to 4, like for the case of IgM relative to IgG. And then also we have oligosaccharide side chains, no? The differences on those oligosaccharide side chains. Okay, antibodies in higher mammals, as I've already mentioned, are enumerated here. Aside from their compositional differences, they also differ in terms of their total size, total charge, essentially because of the chemical complexity. No? And in general, our antibodies, all of these five, no, they may function as follows. They can attack microbes, but do not necessarily they can attack microbes that are free, that are yet to be or are that are yet to infect cells. They can also attack microbes that are exposed in the open, so those that are free floating, not yet stimulated, not yet attracted to a particular host cell. They may also neutralize viruses, and they may also neutralize toxins. The attack functions of our antibodies, they're carried out via several mechanisms such as agglutination. No, so after agglutination, our antibodies that are capable of, or how agglutination happens, we expect paratopes in the antibodies to bind to epitopes of the antigens presented or antigens within the cell, di ba? Then we expect that in agglutination, it's possible that a single antibody can attract more than or can bind to more than one cell or more than one antigen. Then we also have opsonization as another attack form, no attack mechanism. So here, the antigen or the organism will be coated by the antibody, and as such, such coating will enhance phagocytosis. Okay. Then we also have complement activation that will essentially result to cell lysis. No? We'll further talk about that in succeeding modules. We also have inflammation that can be considered as an attack mechanism as well. Attack mechanism in the sense that, as the foreign cells will try to invade or will try to enter the body, we expect that multiple immune cells will be recruited to that region, primarily by the action of antigen recognition, and then we know that antibodies play a very important role in that. No? Now we also have antibody-dependent cell-mediated cytotoxicity. Okay, so antibodies attached to a target cell will cause destruction by non-specific immune cells. Then for neutralization, as an example, is this image here. So we can see that once antibodies cover an antigen, no, instead of if they are not successfully phagocytosed, they may still try to enter the host cell. And what happens is if they are bound or they're covered by antibodies, they're technically neutralized. They will not be capable of uh, doing their effector processes in order to enter the cell. And for the case of toxins, that's also possible. Instead of the toxin being capable of binding to its specific receptor, the antibody will neutralize it. No? The antibody can neutralize it. Okay, so we'll have another 10 to 20-second break. Okay. Okay, so now let's continue with our discussion on the different isotypes of antibodies. Going back to the five isotypes of antibodies or immunoglobulins, the main difference lies on the amino acid sequences in the constant region, as I've already mentioned, of the heavy chain. These amino acid differences can be studied directly through sequencing or also via serological means. You can do serum testing in order to identify what type of antibody is present in your sample. But again, to reinforce the idea, I'm reiterating that the differences, the isotype differences, relies on the subtle complexities that are found on the heavy chain constant regions. Okay. All right. So the five immunoglobulin isotypes, we have IgG, comprising of the gamma heavy chain. So that's the unique type of heavy chain it possesses. IgA, alpha heavy chain. IgM, mu heavy chain. IgE, epsilon heavy chain. And IgD, delta heavy chain. Okay. Now, if we appreciate their visualization in 3D. Okay, so here we have IgG, IgA, the IgM in pentameric orientation, IgE, and IgD. We'll talk about each of them in the succeeding slides. No? But first, let's take a look at the general biology, the general biological characteristics of our Ig molecules. No? So let's examine their molecular weight. IgM is the biggest, the um, heaviest among the five, weighing 900,000 daltons. Then the next is IgA, followed by IgG and IgD. And then, next is next is IgE. So we have IgM, the heaviest, IgA, 360,000 daltons, IgE, 200,000. And the lightest are IgG and IgD. No? Now, this I've mentioned already in terms of number of heavy regions, constant heavy regions, the heavy chain type. Okay, so you remember the smallest one, the least weighty, the IgG and IgD possess three constant heavy regions, CH1 to CH3. Okay. Now, in terms of synthesis area, there are two separate regions. For IgA and IgE, they are produced in the intestinal and the respiratory tracts, while IgM, IgG, and IgD are produced in the spleen and lymph nodes. Okay. Let's examine each of the Ig isotype at this moment. Let's start with our IgG. The most basic one, IgG. So all IgGs are monomers, okay? And there are four subclasses of IgGs. We have our IgG 1, G2, G3, and G4, which will depend on the number of disulfide bonds and the length of the hinge regions. So here, so an example as illustrated here as well. So we have IgG, gamma 1 heavy chain, okay? Then we have IgG2, gamma 2 heavy chain, it's represented here. And then IgG3, gamma 3 heavy chain. So you can see that for IgG3, the heavy chain region is actually, I don't know, longer. Primarily, the disulfide bonds are more numerous compared to the others. And then we have the IgG4, gamma 4 heavy chain. Okay. Now, in terms of their dominance, and you can see they're the decreasing number or the decreasing concentration of each. So we expect IgG1 to really dominate among the others, no? Compared to the other IgG types, IgG subclasses. Okay. IgG is actually the most versatile among all types of antibodies, among all isotypes, because they can carry out all potential functions of the antibody molecules. In terms of concentration, it's also the one that is major or mostly dominant in the serum, comprised of about 65 to 75% in terms of volume. It is also the dominant immunoglobulin in extravascular spaces in areas where you have connective tissue, you have blood vessel, your immunoglobulin G, or IgG, is also the dominant one. Another unique feature of IgG is they're the only isotype that can cross the placenta. So you expect that they have the domain for in the FC region of IgG. You expect a domain specific for carrying out placental transfer. No? So what do you call this? In terms of in terms of placental transfer pala, remember all this. I have to mention that not all the subclasses have the same potential to transfer to transfer via the placenta, to be transferred to the progeny of the mother. No, so not all IgG subclasses are capable of having the equal potential. Okay, so when IgGs bind to antigens, no, there are several downstream potentialities that are stimulated. So first, the attached cell can internalize antigens better. So there's what we call antigenic shape focusing. So that's one possible consequence. As a good opsonin, also the IgG can prepare the antigens for processing by phagocytic cells, and the overall process of phagocytosis is enhanced by the IgG molecule covering the whole antibody. Okay, I mean covering the whole antigen. Now, aside from phagocytosis, aside from better recognition of the antigen, no, antigen focusing, okay, IgG also has other functions. Okay, IgG also fixes complement. They bind to complement proteins in order to initiate the cascade activation. So not as IgGs bind to complement the whole, the whole activation process occurs. Though not all subclasses again will have equal capacities to fix the complement cascade, or to activate the complement cascade. In fact, no one of the subclasses, IgG4, does not fix complement. Okay, cells binding to IgGs also occur not only within phagocytosis but also with cells that have the appropriate FC receptors. So it's not only during phagocytosis that you expect the interaction to happen. Other immune cells, as I've already mentioned, also possess the FC receptor that can be recognized by our IgG. Therefore, that binding can be possible not only with phagocytes, no, but also with some other types of lymphocytes. As a reminder, again, not all subclasses of IgGs bind equally well to cells. IgG2 and IgG4, they do not bind to FC receptors that efficiently. Okay, here are just representation of the mechanisms, the consequences of IgG binding to antigen or to the foreign cell that is being targeted. Um, one thing I have to emphasize is the transplacental transfer. So as transplacental immunoglobulins, what do you call this? IgGs are important for fetal immunity. So the sterility of the environment within the mother's womb. So we expect that early on the development, an animal body is not expected to have um, immunity that is that is already capable of fighting foreign foreign pathogens that are existing outside or in the external environment. Therefore, fetal immunity owes a lot to our IgGs, no? So crucial for fetal immunity normally for 6 months, kasi during this early period of the um, um, newborn, very 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 naive pa yung immunity niya, yung kanyang immune system. So though it might be problematic if we factor in some blood specific antigens like Rh, no, for the case of transplacental transfer of IgGs. Okay. Now, in terms of neutralization mechanisms, neutralization of viruses can be accomplished, when antiviral antibodies block the attachment of the virus to the cells. I'm not going to give too much discussion on this anymore, because I think you have your virology classes as well. Now, we have antibody-dependent cell-mediated cytotoxicity. Now, we have ADCC. So the antibody-mediated, antibody-dependent cell-mediated cytotoxicity, ADCC, is a cytotoxic reaction that's mediated by non-immune effector cells against target cells that are sensitized by antibodies specific to the antigens of that target cell. Those antibodies, they belong mostly to the IgG class. Kaya nga we've made a special mention on how attack mechanisms like ADCC fall under the IgG group, no, and fall under the IgG mechanisms. Several types of white blood cells, they can play a role in ADCC, but and all of them, they bear specific Fc receptors for Fc fragments of the IgGs. And the efficacy of each population of effector cells the efficacy of the whole process will be dependent on the species. Okay, but here we just remember how antibodies like IgGs participate in the process of cell destruction by immune cells and or non-immune cells as initiated by antibody binding. Okay, so next we will have our second isotype of antibodies. We have immunoglobulin A, IgA. Let's have a 10 to 20-second break. Okay, now let's go back to discussing the second type. Now we have IgA. So serum IgA is a monomer. Okay, um, and sorry. So serum IgA is a monomer, and it's highly associated to secretions. So in secretions, IgA are found as dimers with an associated J chain. So here, if it's secretory, sabihin if it's produced by the plasma cell to be secreted outside, then we expect that it will form a J chain. No, it will exist as a dimer, and we'll have that associated J chain. Okay. Um, let's have a quick review on the properties of IgA. Okay, so here. So IgA is the second most common serum immunoglobulin. IgA is a major class of antibodies and secretions, no? As I've already mentioned. And normally, IgA does not fix complement unless it is aggregated, unless that they can be combined into multiple units. And IgA can bind to some cells, especially polymorphonuclears and some lymphocytes. In terms of production, in terms of location of production, activated B cells that produce IgA in secretory format are found in I don't know in mucosal associated lymphoid tissues. And some IgA enters circulation through the lymphatics after they're secreted. Some migrate to the basement membrane of the epithelial mucous membrane so that they can express themselves as dimeric form of the as the dimers or the J chain. So essentially you divide your pool of serum of IgA into two. Meron kang those that are those that go back to circulation and those that stay in the in the connective tissue, stay in the associated lymphoid tissue. Okay, the secretory IgA, mucosal secretions such as mucus, tears, saliva, colostrum in the blood of a mother, no? So they all possess trace amounts of serum IgA, no? And um, as a secretory immunoglobulin, they're important for local immunity. No? They're important for local community kasi they're found on specific secretions. Therefore, the secretions on the site of their secretion will essentially depend, the immune process will depend um, um, partly on the present IgA. Okay, so in mucosal regions where they're immediately available, they can immediately act as neutralizing antibodies that will prevent any foreign agent from attaching to the cell. Okay. All right, so the third isotype is the IgM. So we're done with IgA. Something you have to remember about IgA again, secretory. Now for IgM, IgM is the third most common serum Ig, and it's normally existing as a pentamer, but it can also exist as a monomer. I've shown you the illustrations a while ago, so you can see it appears in that format, no? Para siyang pentagon yung appearance niya. IgM is also the the first immunoglobulin to be made by the fetus and the first immunoglobulin to be made by a virgin B cell when it is stimulated by an antigen. So it's one of the initial immunoglobulins that are produced, IgM. Let's take a look at the pentameric nature of our IgM. Let's have here photo. Okay, so you can see also once that you have monomers of your IgM that are oriented in this pentagon-like, or this is actually pentagon in shape, no? So once that they are oriented in this pentagon conformation, okay. So here we can see that there's a presence of the J chain. So we first recognize or identify associated with our with our secretory IgA. Okay, and if we take a look at the heavy chains and the L chains of pentameric IgM, they're all identical. Okay, so the pentameric nature also provides us a lot of things to ponder about the behavior, about the activity of IgM. In IgM, as a consequence of the pentameric orientation, we have a few things that can be carried out by the IgM. It's a good complement fixing antibody, very efficient in leading to cell lysis by means of complement activation. No? Primarily because of its, marami kang marami tayong antibodies. No, there are more than one or more than two. No, it's not only a dimer but a pentamer. IgM is also um, it's also good in terms of agglutination, primarily because of the structure. It can help in clumping microorganisms for succeeding elimination from the body, primarily by phagocytosis or other means of foreign pathogen elimination that is antibody dependent or antibody initiated. Okay. Then also IgM can bind to some cells using the Fc receptors. Okay. Now, if you take a look at the monomer of each IgM, a monomeric IgM can be found on as an example, an IgM working as a BCR, as a B cell antigen receptor. No? As monomers, they're membrane bound and they can function as B cell antigen receptors. Their proper orientation in the cell membrane of the B cells can be attributed to their having non-covalent associations with two additional proteins that are represented in this picture. Okay, so the Ig alpha and the Ig beta proteins. Both of these proteins are signal transducing molecules. They serve as initial elements of signal pathways where our IgM as a BCR is involved. No? So all signal transduction molecules or all signal transduction pathways where your IgM is involved, we expect that we expect what they call this. We expect these two associated proteins, the Ig alpha and the Ig beta, to be highly involved. Okay. Next we have so contact between B cell surface IgM and an antigen. It's actually the primary. I don't know. It's a primary. What do you call that? The primary event. No? In order for signal transduction to proceed, there should be your IgM must recognize an antigen. So once that recognition occurs, or once the recognition has has transpired because of the contact, then we expect signal transduction via the succeeding protein, especially the Ig alpha and the Ig beta chains. Okay. Now, B cell that is activated because of that antigen recognition will differentiate into an antibody secreting plasma cell, and in some instances, okay, um, you expect that the secretion of our IgM will what do you call this, the sorry, something I have to mention here. Okay. Yeah, okay. So I mentioned it already. So yeah. We just have to remember yun, yeah, as a a B cell antigen receptor, we expect IgM to uh, be an initial structure point, no, for, or as an initial structure for, what do you call this, for signal transduction. And I'm telling you, a lot of signal transduction routes can be activated once that your IgM is bound to the specific antigen it carries. Okay, next we have immunoglobulin E. Before we proceed to this, I'll have another 10 to 20-second break. Okay, so now let's go back to our discussion and let's describe what IgEs are. So immunoglobulin E, or IgE. The second to the last type of Ig that will be covered in our in this preliminary lecture. So IgE, they exist as monomers and they have an extra domain in the constant region. IgE is the least common serum Ig since it binds very tightly to Fc receptors on basophils and mast cells. Probably you've always encountered mast cells and basophils with IgE. No? Most of the time they exist in the same sentence. If you've been reading, most of the time that you've been reading about, about, what do you call this, um, about what do you call this, allergic, mostly no, allergic reactions, and you can encounter IgE together with mast cells and basophils. So they're least common in terms of serum prevalence because we expect that they are tightly bound to the Fc receptors of mast cells and basophils, and IgE does not fix complement unlike the others. Okay, so here we can see the representation of IgE. No? In allergic reactions, histamine is uh, histamine, is actually involved, or IgE is involved in allergic reactions. Once that there's binding of allergen to the IgE on basophils and mast cells that will result to the release of various pharmacological mediators like histamine, and those substances mediate allergic symptoms. Each type of IgE, it's known that in each type of IgE, they have specific radar, code, and for each type of allergen, specific IgEs may have their radar. So kaya nga meron tayong differences ng allergen potential or allergen what do you call this profile. Not all people will be allergic to the same allergen, okay? Because of essentially the differences in the nature of their IgEs. Okay, so each IgE that you will have, that you will produce, will have that kind of specificity. Okay, so um, here let's take a look at a quick overview of an allergic reaction and how IgE is involved in it. Yeah. Okay, so we start with an initial contact with an allergen. So our allergen being recognized by our IgE on the B cell. Okay. Sorry. Let me no. Let me clarify. Okay. So yeah. Let me go back. So again, let's start with an initial contact with the antigen, with the allergen serving as our antigen here. So our IgE bound to B cell, capable of recognizing that allergen because of its specific, specific radar for each type of allergen, right? Then activated plasma cells will secrete IgE. Okay, IgE released as antibodies on the serum. They will attach to mast cells or basophils, because the mast cell and basophil have specific Fc receptors for our IgE. And then once that there's subsequent contact with the same allergen, succeeding contact with it, we expect mast cells or the ones that mast or basophils to degranulate in order for them to release histamine and other pharmacological components that will essentially comprise or will create the allergic reaction process. Okay. In aside from allergy, or aside from the allergic reaction, IgE is also involved in parasitic infections. No? So IgE in parasitic infections, the Fab portion, the fragment antibody binding portion of the IgE, it reacts with epitopes on the helminth, while the Fc portion will bind to the Fc receptors of activated eosinophils. So I've made a mention about eosinophils before, diba? So eosinophils as important cellular structures or cellular types for parasitic, for helminthic infection. No? And being two-way, since serum IgE levels rise in parasitic diseases, therefore, we can measure IgE levels as a helpful indication, no? As a diagnostic tool in checking if parasitic infections are present or not. So here, so this is just what I mentioned a while ago. No? So we expect how your IgE will be helping in the elimination of parasitic infections, especially helminthic infections. Okay. The last antibody isotype that we will be covering in the lecture are IgDs. No? IgD functions mostly as a receptor, a B cell receptor. And few of its characteristics, let's review. We have IgD existing only as a monomer. Okay, IgD found in low levels in serum because, mostly acts as receptors. In fact, until now, no, medyo hindi pa ganoon well thoroughly studied ang IgD as a type of antibody. Okay. So also it's as a mostly cell membrane bound No, functions as a receptor for antigen, and also it does not bind complement, does not fix complement, unlike the other types. No? Within each of the ano na, no, because I'm just going to make a quick mention of the others na like IgD kasi ano naman siya, very very known pa yung alam natin so far. No? Aside from or within each what do you call this antibody isotype, greater heterogeneity can be observed. No? So aside from the isotypes, as you know, offering us a look at the diversity of the different antibodies within each isotype. Okay, there's greater complexity, greater diversity. Light chain types may exist. There are what we call kappa and lambda types. Heavy chain classes may also exist. Like for the case of what they call the subclasses of polypeptide chains for IgG, for IgA, and for IgM. Then we also have allotypic configurations of the heavy and the light chains, which has something to do with alternative structures based on genetics. Okay, so in summary, yan, we have these five isotypes of antibodies. We discussed them so much, and I hope now we are able to appreciate how biologically different they are from one another, their genetic functions, and the things that they share with one another, the features that they share, the features and functions that they share with one another. Okay, now we are going to proceed with our last section, which will be talking about antigen and antibody complexes. But before we do that, no, let's first take a look at the genetic control of immune responsiveness. Because we've mentioned about how immunoglobulins on the antibodies as humoral effectors participate in the process of immune response. Then we also have our immune cells and our non-immune cells. So in essence, we have genes that are responsible for the interaction between these humoral and cellular components. So we have our cell interaction genes and our immune response and immune suppression genes. No? So this is a generic categorization. Cell interaction, immune response, immune suppression genes, generic sila. All right. Now, to describe them simply, I have some examples here. For cell interaction, so some cell interaction genes are capable of controlling macrophage lymphocyte interactions. So if you remember how T helper cells can participate or can help in can help in macrophage phagocytosis or can help during macrophage resting, macrophage not being capable of degrading the phagocytose antigen. Lymphocyte substances are produced in order to activate macrophages. That's one example. And cell interaction genes are involved in that association. Then we also have interactions among lymphocytes. That's carried out by gene interaction genes, or I mean cell interaction genes as well. No? For the case of cellular lysis, no? For natural killer cells, cytotoxic T cells, no? Their activity is also determined by effector molecules that are produced by means of effector molecules that are coded by cell interaction genes. Okay. Then for immune suppression naman, we have immune suppression genes and immune response genes. They have for immune response genes, they have the function of determining the ability of the individual to respond to a given antigenic determinant. Kaya your immune response genes, because essentially they're genetic, therefore not all people will have the same level of immune response. Okay, and then we also have immune suppressor. So for immune suppression genes, they're responsible for deactivating pathways such as what do you call this secretion of a secretion of humoral factors. They can immune suppression genes may be activated in order to regulate the process. Okay. So, as a summary nga, no, we have this what do you call this gene types. Okay, they participate in the whole in cell to cell as well as immune regulation. Okay, and in our succeeding modules, we'll make special mentions of particular genes, particular genes that are involved in cell to cell interaction or immune activation, immune suppression. So the last topic for our module are antigen antibody complexes. Okay, so before we proceed, I think I only have one, two, three, four slides. No? Actually, after this last slide, I will show some videos, no, on each of the technique that we use to study antigen antibody complexes. Let's have another 10 to 20-second break. Okay, so now let's finish our module number 4 on our discussion about antigen and antibody complexes. So since the earliest times that we discovered antigens and antibodies, no, especially when scientists, immunologists discovered the existence of antibodies, no, and they are defined as important components of the immune system. No? We went to the whole idea, or the whole scientific community went to the journey of identifying, understanding the interaction between antibodies and their antigens. Aside from become aside from being an important tool in understanding um, antigen antibody interactions, antigen antibody complexes help us in understanding the basis of the molecular recognition between the antibody and its specific antigen. So aside from, the interaction itself, the complex that can be normally studied outside the animal body is also helpful in understanding how the interaction comes to be. Kasi we can, after visualization, after really reconstructing the complex, we can see what components are interacting with one another. Okay, and with the aside from interaction, um, we also can infer other information like biological activity. So the biological function specific antigens or proteins can be explained on how they secrete how they initiate immune response. Diba? So as an antigen binds to an antibody, okay, we expect that immune responses will be varied, and the immune responses will be carried out. In an in vitro setup, if we uh, if we will be able to, let's say, for example, successfully isolate the antigen antibody complex that activated that specific immune mechanism or immune response, then uh, we actually have direct evidence, diba, on how antigens or proteins that are foreign to the animal body can stimulate immune responses. For the case of allergic reaction, the case of let's say, for example, a viral infection. Diba? Antigen antibody complexes are very useful, not only in studying the interaction itself, but also in the attempts to understand how the interaction behaves, the entire biology of the interaction, and also how each component participates in the process of interacting with one another, and also in the process of mounting the immune response. Okay. Early on the study, perspectives on antigen antibody interactions have actually changed over time. Ano na siya, very nag-evolve yung understanding natin on how antigens and antibodies interact with one another. So initially, what the idea that is bought, the idea that is sold, is that it's believed that the antigen antibody interactions are described as lock and key lock and key systems, no? So you remember from your biochemistry yung lock and key, Fisher lock and key model, diba? However, as more structural data became available, we expected that it became clearer that in many cases the lock and key model is not properly describing the antigen antibody interaction. Now that change in conceptualization gave us the I don't know, it gave us the mechanism of induced fit. So the induced fit model, it provided a significantly better picture of how antigen and antibodies bind to one another. So as demonstrated by multiple structural studies that were conducted, the induced fit model was made possible due to antibody flexibility, because of the fact that antibodies are highly flexible. You remember the hinge region, diba? And also the conformations necessary in order for an antibody to achieve a specific shape or a specific conformation, diba? So being a highly flexible molecule, yun nga, later on we accepted more the idea of induced fit instead of lock and key. Okay, as since these are protein protein interactions, basically proteins are highly dynamic, and in normal physiological conditions, they undergo continuous conformational changes involving both the interacting chains or interacting residue and the side chains. This dynamic nature of proteins give us the idea that in solution, immunoglobulins adopt various conformation, and there is a pre-existing equilibrium between various conformations of those molecules. Kaya nga, induced fit siya. Kasi you have different conformations of the antibody based on, the physiological conditions of the solution, and as an antigen interacts with it, it will assume the conformation that is most applicable to that antigen. Okay? Can we say that epitopes are also flexible? Kasi we've given so much thought about how paratopes in the antibodies can assume different orientation because of the structural flexibility of antigens, diba? In a way, we can say that, or in a way, epitope flexibility can be viewed, because can be viewed in different light. Kasi you remember cross-reactivity a while ago. Diba? That's a different phenomenon. But in essence, since epitopes or antigens are proteins, and the amino acids are, and the epitope essentially is just a small structure that might be composed, or that is composed of key residues, it can be, no? It's possible that epitopes are also as flexible as paratopes found in antibodies. Okay? Now, let's take a look at antigen and antibody complexes in human health. The binding of antigen to its specific antibody changes the properties of the bound antigen, leading to numerous immune responses, including antigen presentation, inflammatory responses, receptor signaling, Okay, those are a few of the consequences of antigen-antibody interaction. No? Now, with modern day medicine, we exploited the idea of this event of antigen antibody interaction. We exploited this idea to innovate drugs, to carry out immunotherapy, and other biomedical technologies, not only for cancer, for autoimmune disease, but also for autoimmune conditions, and also for cases in which antibodies are depleted or not yet produced in substantial amounts, or in cases in which the immune system is still naive, like for the case of babies, like for those that are inside the mother's womb, the fetuses. Okay. Now, if we take a look further, no, if we examine the role of antigen antibody complexes in human health, let's make a few examples, or let's discuss a few examples. In human health, the association of the antibody with its specific antigen, leading to the effects that I mentioned a while ago, okay, might be beneficial or might be detrimental. Let's take the case of HIV and cancer as examples, AIDS and HIV. I mean, AIDS and cancer, rather. So, the use of antibody complexes in vaccine development and immunotherapy for these two diseases, these two pathologies, has been promising so far. In one example study of HIV on mice models, antibodies recognizing the V3 region of the envelope protein, of the GP120 protein, elicited production of strong neutralizing antibodies against the virus. In cancer and tumor biology, or cancer in tumor therapy, as an example, one common method of treatment is targeting surface factors that are crucial for tumor cell growth, essentially antigens, diba? When antibodies recognize and bind to these antigens, to these factors, example of such factors include epidermal growth factor, vascular endothelial growth factor, VEGF. They form antigen-antibody complexes, and ADCC can be triggered so that tumor lysis can be carried out. Yes. On the beneficial side, that's what antigen antibody binding or complexation does. On the negative side, the formation of antigen antibody complexes has been linked to the development of autoimmune diseases and allergic diseases as well. A prominent example is the major role that is played by IgE in type 1 hypersensitivity. IgE is implicated, diba, in hypersensitivity reactions stemming from the presence of allergens, food allergens, allergic asthma, in the animal body. In allergic diseases, antigen presenting cells present the antigen to T helper cells. The activated T helper cells will now release humoral factors, interleukins like IL-4, IL-13, which will lead to the upregulation of IgE production. So parang ano lang siya, positive cycle siya that will continue, or positive feedback cycle that will continue to proceed. And this antigen-specific immunoglobulins, our IgE, will attach to the antigens will specifically attach to the IgEs. And then the cycle might be negative in the sense that it will continue to proceed to proceed, and it might be problematic because if hindi ka magkaroon ng immediate ng first aid in such hypersensitivity reactions, it can be detrimental, it can be lethal. Okay, so we see how antigen and antibody complexes can participate both in negative and positive interactions, or negative and positive events for human health, now for in certain human pathologies. Okay, now how can we study antigen antibody interactions or antigen antibody complexes? Okay, there are several methods or several techniques. So we have X-ray crystallography, we have nuclear magnetic resonance, and then we also have single particle cryo-electron microscopy. So let's take a look at each of them before I show you some videos. So first we have X-ray crystallography. Since most B cells, the most of the antigens that bind to um, B cells are conformational as well. No? X-ray crystallography can be considered as the gold standard in studying three-dimensional or producing three-dimensional structures of antigen-antibody complexes. X-ray crystallography is able to produce a snapshot of the antigen-antibody complex at high resolution, and it makes it possible for us to visualize the specific interactions between the antigen and the antibody. However, because antibodies are immunoglobulins or having the immunoglobulin domain, medyo ano sila, no, bulky sila. It's hard to crystallize them. And and an initial thing in X-ray crystallography is crystallization. No? So we will crystallize our complex first before we observe it so that we can determine the X-ray structure, the X-ray crystal structure. Let's have here one video. Okay, how X-ray crystallography works.
If you want to determine the structure of a protein by X-ray crystallography, you first have to grow a protein crystal. Unlike table salt, proteins don't crystallize easily because of their irregular lumpy surfaces, so numerous attempts may have to be made to make a crystal. Various factors, pH, temperature, the solvent, are important for success. Salting out agents such as ammonium sulfate can reduce the solubility of the protein to aid crystallization, but it may take several days to produce a crystal large enough to use. The protein crystal is mounted in a specific orientation and exposed to a beam of X-rays. The atoms in the crystal scatter the X-rays, which are recorded on film. Each atom in the sample scatters X-rays, and the strength of scattering is proportional to the number of electrons in the atom, so each atom can eventually be identified. Since you can't focus X-rays with a lens, a different method is used, a mathematical formula called a Fourier transform, which can interpret the X-ray photograph to produce a contour map of electron densities. This map represents the electron density for a particular plane through the crystal. The peaks and valleys correspond to the positions of atoms. Here is the heme in hemoglobin, whose atoms all lie in the same plane. The hill in the middle is an iron atom. Since proteins are three-dimensional, many such maps, each representing a slice through the molecule, must be assembled by computer to create a complete 3D structure.
Okay, so that's a short video showing us an overview of how X-ray crystallography works. Okay, now let's proceed to the second type or the second example of a biological method to study antigen-antibody complexes. We have nuclear magnetic resonance. So NMR is a method that is able to map residues forming epitopes and paratopes together, and it allows to provide, or it gives information on intermolecular interactions. The biggest advantage of NMR over X-ray crystallography is there's no need to produce a crystal. So it usually is carried out for those proteins that are hard to crystallize. The basic idea for NMR is that um, in NMR, epitope mapping and antigen-antibody interactions, um, amino acids on antigen binds to an antibody, the amino acids chemical environment will change. The chemical environment of residues on the antigen-antibody interface changes because of the protein-solvent interactions that are replaced by protein-protein interactions. Now, the NMR signal the NMR signal will be able to um, um, or the NMR equipment will be able to measure that chemical that signal change, and the interaction between the what do you call this between the atoms and molecules can be mapped using those chemical signal changes. Okay, we will be showing a video in order to further discuss, the technicalities of nuclear magnetic resonance. Let's take a look.
Nuclear magnetic resonance is a technique that uses magnetic fields and electromagnetic frequencies to study molecular structures. For high magnetic fields, superconducting magnets are used. Lower fields can be generated by permanent or electromagnets. Samples are inserted in the center of the magnet and are positioned in the NMR probe. So the sample is located at the position of the strongest field and highest homogeneity of the magnet. The NMR probe consists of coils that are used to excite the sample and record signal responses at radio frequency. The basis of NMR is that nuclei behave like microscopic magnets in a magnetic field. They spin around the main magnetic field just like a spinning top. This motion is called precession. The frequency of the precession is proportional to magnetic field strength. Additional local field differences are introduced by changing electron densities in a molecule. A higher electron density results in more shielding and thus a lower local field and lower spinning frequency. In NMR, the frequency of the excited nuclear spins can be measured from the electric current they induce in the coil. Fourier transformation of the time domain signals results in the frequency spectrum, with a signal for each different nucleus. The different frequencies we call chemical shifts, as they depend on the chemical structure of the nuclei. In practice, spectra are measured for all hydrogen nuclei at once, which results in the ethanol spectrum with 3 different peaks at different chemical shifts. Since nuclear spins behave like magnets, they also influence their neighbors. One way this happens is through shared electrons in the chemical bonds in the molecule. We call this J-coupling. Each spin can have 2 states, along the magnetic field or against the magnetic field. As an example, we look at the 2 methylene hydrogen spins, which are chemically identical. They can have 4 combinations: 1 spin down, 1 up, both spins down, 1 up, 1 down, 2 spins up. Each combination has a different energy level and influence on the neighboring spins, except that up-down and down-up combinations have the same energy level and influence. The influence is visible as a splitting of the signal of the methyl group into 3. This is called a multiplet. Likewise, the 3 methyl hydrogen spins split the methylene response into 4 signals. In conclusion, NMR can identify molecular structures from chemical shifts and multiplets.
Okay, so as applied in studying antigen antibody interactions. So yung NMR, the process, the technique, nuclear magnetic resonance, what we do is we study the differences in the NMR spectra before and after an antigen is bound to an antibody. So that's how we use NMR in structural studies of antigen antibody complexes. So you subject your antigen to NMR without the antibody yet. Then afterwards you add the antibody to the antigen, allow them to complex, and then you subject it again to NMR so that you can identify the differences in chemical shifts. And with such, we can identify what interactions are happening or what molecules, to be exact, what atoms are interacting between the epitope and the paratope. Now, the last technique that we enumerated here is cryogenic electron microscopy. So this technique is applicable in studying very large molecules. such as viruses, bacterial appendages, eukaryotic ribosomes, cellular organelles, and for the case of antigen-antibody interactions, very much applicable as well. Let's take a look at how cryo-electron microscopy works here.
Proteins are the building blocks of our cells. Understanding their shape and how they fit together can help us treat pain and even cure diseases. Scientists have been trying to get a good look at some proteins, but for years, the best we could see was a blurry blob. Many proteins are thousands of times smaller than the width of a human hair. Traditionally, scientists use a technique called X-ray crystallography to take pictures of the tiny proteins. For the X-ray imaging to work, it requires proteins are packed together into a stable, organized crystal. X-rays pass through the sample to create an image, but some of the body's most important proteins are too floppy or wiggly to line up into a crystal. Scientists at UCSF have developed a better way to view these elusive proteins.
If you have a floppy molecule at all, they just become more and more challenging to crystallize. Really, the problem was that these are big, complicated systems, and coming up with new ways of studying them required thinking about electron microscopy.
The reason cryo-EM works is that we keep the protein in a very thin layer of liquid, and we freeze them.
This technique is called cryo-electron microscopy. An electron gun shoots electrons at the speed of light passing through the sample. Then a specially designed high-tech camera captures the electrons to form an image.
The camera not only improves the imaging condition, give you much better sharp image, it can record a movie instead of a steady picture.
If you had a still picture and I was doing this with my hand during the picture, I had a blurry hand picture. But with a movie, you could actually see each of the different steps.
A computer algorithm sorts the images and finds pictures of the protein in the same orientation. Then software builds a composite, an accurate high-resolution, three-dimensional image of the protein.
And we're using the computation method to average many hundreds of thousands of molecules or tens of thousands of them together to generate a three-dimensional structure.
A structure like this one. Yifan calls it the wasabi receptor because it detects the pungent zing in wasabi and onions, and it's part of how the body registers pain. Scientists have known about this protein for years, but now we can finally see its shape in 3D. That means we can begin to build better drugs to block pain.
Okay. All right, so that showed us a very concise explanation of how cryogenic electron microscopy works. So for the case of all these three techniques, we see how they are heavily involved in determining the structure of the analyte of either the antigen or the antibody. But for our special case, how the structure of the complex, the antigen and antibody, how the antigen antibody complex interacts with one another, especially the involved atoms, the involved molecules within the epitope and the paratope. Okay, so that concludes our session for module number 4. Then for module number 5, we will be, since this falls under the Lasallian Festival week, so we will be holding the classes, and instead what will happen is, what we will be doing is, what I will be doing is that I will also record a series of lectures for module number 5 on T cells and B cells. So expect this almost the same format. We're going to record it again, and I will make the PowerPoint discussion or the slide deck available as supplementary material. Okay? Thank you.